Hi, welcome back to the Linterman Lab YouTube channel. I'm William Foster and today I'm going to talk you through our recently published paper which has come out in Immunology and Cell Biology. The paper is titled ChAdOx1 nCoV-19 vaccination generates spike-specific CD8 T cells in aged mice. This paper was a collaboration between our lab and the labs of Theresa Lamb in Oxford and Dalen Bailey at the Purbright Institute. This will be a short video in which I show you the figures of the paper. I provide you with the key messages, so if you don't have time to read the whole thing, you can just watch this video. In figure 1, we start off by comparing a cohort of young, which are 3 months old, and aged, which are 22 months old mice. We immunize them with ChAdOx1 nCoV-19, and then 6 weeks later, 42 days later, we looked at their serum. In A, we show a pseudo-neutralization assay, and the old mice have a reduced ability to neutralize viral particles using their serum. In B and C, we perform two sets of ELISAs. B is an anti-RBD ELISA and C is an anti-spike ELISA. For B, we see that the old mice have a reduced amount of anti-RBD IgG in their serum, and for C, we see a 0.07 trend for reduced anti-spike IgG. In D, we performed linear regression analysis where we compared the neutralization capacity on the x-axis to the anti-spike IgG ELISA data on the y-axis. We performed the statistical test for the young cohort and the old cohort separately, and for both there was a positive significant correlation between our two metrics. So this suggests that if you have an increased amount of serum IgG for anti-spike antibodies, then you'll have an increased capacity of neutralization. So the message from figure 1 is that aged mice have a reduced serum humoral immunity following their ChAdOx1 nCoV-19 vaccination. And this correlates with several human data sets which have been published, suggesting that the older you are, the more susceptible you are to SARS-CoV-2, even if you've been vaccinated. So this is really important. But this is not the only method by which you can be protected from viruses. Another method is by CD8 T cell protection, and so we went on to focus on this on the rest of the paper. In figure 2, we begin looking at the CD8 T cells. 2A, we define our CD8 T cells by flow cytometry using a viability dye, looking at forward and side scatter, and then using CD4 versus CD8 staining. In figures 2B and 2C, we did a time course study where we immunized mice with either the ChAdOx1 nCoV-19, this is the black plot shown on the left. We also immunized mice with ChAdOx1 OVA, this is the same vaccination backbone but expressing a different protein, the OVA protein. And we also immunized mice with PBS, so this is the liquid carrier in which the two other vaccines are suspended in. And what we see is that only mice immunized with the ChAdOx1 nCoV-19 generate spike-specific CD8 T cells. And so in the flow cytometry representative plots, you can see that we stained the cells with two different tetramers. These are MHC class 1 tetramers, which hold a piece of spike epitope on two different colors. And then we find the dual staining cells, and these are the spike-specific cells which have been produced by the vaccine. The graphs in C will show you that both in the medial iliac lymph node, this is the draining lymph node for our vaccination site, and also the spleen, we see spike-specific CD8 T cells being generated. Another important note from this figure is that the number of spike-specific CD8 T cells that's in the spleen far outnumber the number which are in the lymph node. This is because the spleen is a larger organ with more total cells, but also because the frequency of tetramer-positive cells in the spleen is higher than in the lymph node. So now we've laid the groundwork and we've shown that the vaccine really nicely generates spike-specific CD8 T cells and that we can find them by flow cytometry. So we went on to then compare the young and the aged mice. The young mice are the black plots with the black dots on the graphs, and the aged mice are in red. And what we found actually is that in the lymph node, the aged mice produce more in terms of a frequency of total CD8 T cells, but also more in terms of total number of spike-specific CD8 T cells than the young mice. And in the spleen, the numbers are broadly comparable. And so this suggests to us that actually the aged mice are capable of producing spike-specific CD8 T cells, and that aging has not damaged this process. In figure 3, we went on to look at the phenotype of these cells because the phenotype can give an indication of their functional capacity. And so on the left-hand side of your screen, you've got A, C, and E, and these are the spike-specific CD8 T cells. And in the middle of the screen, these are the B, D, and F panels. These are the total CD8 T cells. So starting off in A and B, we look at CD44 expression versus CD62L expression, and this gate shown identifies the T effector cells. The majority of the spike-specific CD8s are of this T effector phenotype, and the aged mice have more of this phenotype than the young mice. In C, we looked at PD-1 expression. PD-1 is an activation marker. It can also be considered an exhaustion marker in certain circumstances. And we see that the aged mice have a higher frequency of PD-1 expression in the spleen, both for spike-specific and for total CD8 T cells. In E and F, we looked at TBET and we looked at CXCR3, and the dual expression of these two proteins has been described as markers of activation. You can see that practically all of the spike-specific CD8 T cells are TBET and CXCR3 dual expressors. And when we look at the total CD8 T cell pool in F, the aged mice have a much higher frequency of this dual expression. This data is all in the spleen. So then in the next figure, we did the same analysis but looking at the lymph node, and our results were largely the same. We found that the majority of cells, again, are of this T effector phenotype, CD44 high, CD62L, although actually in the lymph node there was no difference between the spike-specific CD8 T cells for young and old mice. However, when we looked at the total CD8s, we found slightly more CD44 high, CD62L T effector cells in the old mice. Looking at PD-1, we saw more PD-1 expression on the spike-specific CD8 T cells and again on the total CD8 T cells. And looking at the activation markers of TBET and CXCR3, we saw that basically all of the cells are TBET and CXCR3 dual positive, so there was no significant difference for the spike-specific cells in E. But when we looked at the total CD8 pool in F, the aged mice had more of these activation markers. So the key message is that aged mice can still produce spike-specific CD8 T cells after being vaccinated with ChAdOx1 nCoV-19. They do have this altered phenotype, and so the question is, can we activate these cells? Because if they could be activated successfully, then that's one method by which you could provide immunity. And this would be by clearing virally infected cells, and this is doubly important because in the aged mice, as we've seen, humoral immunity is reduced. If you'd like to know more about the paper, please send me an email. You can also contact me on my Twitter and on my LinkedIn. And if you'd like to watch the previous YouTube video that we uploaded, you can do so on the links shown here. Thank you to our funders, the BBSRC and the Lister Institute. If you like this video, please give it a like and subscribe to our channel if you'd like to see more video breakdowns of our upcoming papers. And please share it on Twitter if you're able to do so. Thank you.